Hi friends, you're back with me, Professor Girish Kukreja. Right. So today we'll be quickly having an overview about finding out the concentration of the DNA. Many of the times when in a molecular biology lab you are isolating the DNA, and once you isolate you want to know how much it is, right? So we are interested in finding out the quantity of DNA. Now there are different methods. One of the most commonly used method in a any laboratory where we have a UV visible spectrophotometer is the absorbance at 260 nanometer, right? So we all know that we use a quartz cuvette. You can go somewhere else in the same channel and find out what are the different ways of estimating this DNA in the same channel you will find. Anyways, so I have the purified DNA solution, say for example, and I took that sample and I took the absorbance at 260 nanometer. Oh, it's not kilometer anyways. So at 260 nanometer it is 0.65, right? In a quartz covered, obviously, in a 1 centimeter path length. So there are some things which we assume, but here I thought that let us not assume the things, right? So it was a quartz covered, thank God the path length was 1 centimeter. I want to find out the concentration. Quickly, you tell me when it is an absorbance of 260 nanometer and the absorbance is equal to 1, we know that the concentration of a double stranded DNA is 50 microgram per ml. So in this problem also it was assumed that you know this. But then I thought that some might not know it, so I thought that I'll tell it, right? So our absorbance of 260 nanometer of 1 corresponds to 50 microgram per ml. So we have that same formula, C1 upon A1 is equal to C2 upon A2. So my concentration of the standard is 50, my absorbance is equal to 1. Then we have the C2, right? So it is concentration I want to find out. Then we have A2, that is 0.65, right? So my concentration is 0.65 into 50, that is 32.5 microgram per ml, right? My mathematics is so fast. So basically whatever absorbance you get, to cut it short, whatever absorbance I get, let me multiply that particular absorbance with 50 and I'll get my concentration. Great. So that is a better version now. So whatever absorbance you get at 260, now you take that and multiply by 50, so that will give you the concentration. Now concentration of the DNA is known, I have a concentration of 40 microgram per ml. And now I want to find out what would be the absorbance there, right? So sometimes the one thing, you want to find out the other thing. I want to guess what is the absorbance. So the concentration of DNA is 40 microgram per ml, right? Now I want to find out the absorbance. So I'll go with the same thing, right? Now with the same thing I have C1 upon A1 is equal to C2 upon A2, where my C1 is 50, absorbance is 1. Now the thing is that I know the concentration, which is 40 upon, I don't know the absorbance, so it is A2 is 40 upon 50, right? Can you solve it out for me? So that's very easy. I request you to solve it. Yeah? You're solving it? Okay. Free. Done? Yeah. So this absorbance is 0.8. So absorbance at 260 nanometer of 40 microgram per ml would be 0.8. So simply here you remember that if I know the concentration, now this concentration divided by what is called as 50. So if I know the absorbance multiplied by 50, I get the concentration. If I know the concentration divided by 50, I'll get the absorbance, right? So a lot of stuff with DNA and all. So stay tuned with me, Professor Girish Kukreja, for more in microbiology, biotechnology and biochemistry. Thanks a lot.